Welcome to the annual update for the BBSRC Follow-on Fund. The fund is our flagship program in translational research, where we invest to progress bioscience discoveries into real-world impact.
Over the next hour, we'll be exploring the goals of the fund and how we invest. We'll showcase some examples of successful completed projects and present an overview of the new awards made in this year. The Follow-on Fund is a strategic investment in bioinnovation made by the BBSRC. BBSRC is the Biotechnology and Biological Sciences Research Council, one of the 9 bodies that make up UKRI, which is the UK's research and innovation agency that invests over £8 billion each year across every part of the UK to create new knowledge for economic growth and to benefit society. BBSRC is funded by the public to unlock the opportunities presented by research in the biosciences. Our strategic delivery plan describes our vision for bioscience to contribute to a healthier, more prosperous, and more sustainable future. The plan also describes how we will achieve that vision by building and strengthening pathways for the translation of discoveries into impact. Since 2004, the fund has invested approximately £80 million in more than 500 projects to drive bioscience discovery through to outcomes such as products, services, spin-outs and licensable IP that has benefited the UK both economically and socially. Awards take the form of a UKRI commercialisation grant, and eligible researchers from UK universities and institutes are able to apply for up to £800,000 to advance their idea. The fund makes awards twice each year, with opportunities opening in both the spring and summer. The Follow-on Fund is a strategic investment in bioinnovation made through the BBSRC's Knowledge Exchange and Commercialisation Unit. In the 20 years of its operation, the Follow-on Fund has provided specialist support that has enabled bioscience research to be translated into a variety of impacts. This slide has short examples that highlight the breadth of bioinnovation supported by the fund. Projects have included field trials to demonstrate the efficacy of agri-tech and to secure the continued production of economically significant crop species in the UK. New collaborations to bring rapid diagnostics for animal health to market, novel food processing technologies that have led to new product launches, proof of concept work for novel drug discovery platforms that have generated patents, prototyping of devices to improve patient monitoring, leading to the formation of a new company, de-risking the development of novel therapeutics to open entirely new treatment options and attract private investment. The fund has a demonstrable track record of enabling discovery research to progress into innovations that unleash the potential of bioscience. We'll now move to a case study presentation to explore the difference that follow-on funding can make to your project in more detail.
Hi, I'm Caroline Barelle and I'm delighted to get the opportunity to tell you a little bit about a project that was funded by a follow-on fund back in 2012. And it focused on our Solamer technology, particularly a domain called EO6 and its ability or potential ability to extend the half-life in serum of multiple other therapeutic drugs. So the overarching aim was to use this funding to produce a data-driven evidence package that enabled EO6 to be clinically developed. So looking at the project as a whole, we're very much interested in biologics, particularly antibodies. And what we've developed is a unique antibody-like technology platform. We call these domains Solamers. They're single chain, they're small, they're robust, and importantly, they bind into targets in a very different way from antibodies, often eliciting a new mechanism of action, which is a great differentiation factor when it comes to making a difference in the clinic. This particular project was focused on something we call a biotool. So a biotool is really important. It enables you to do something in the clinic that other domains can't do. And in this case, using EO6, which binds human serum albumin, it can extend the half-life of other drugs for longer. Why does that matter? Well, it matters from the perspective of developing drugs for chronic autoimmune diseases, for example. it decreases the dosing and increases the quality of life for patients who are being subjected to these drugs, often early on in life and for the rest of their lives. So a little bit about the Solamer technology itself. So as I've said, these are single domains. They are in fact shark derived, which is fascinating. These beautiful animals also have other antibodies in their body, but these ones are 10% the size of classical antibodies. And because they're single chain, they have the capacity to be able to be linked together. So really pushing forward the limitations of other antibodies in that they can bind multiple different targets at one point in time, which is really important for complex diseases. And here you can see on the left-hand side, we're developing autoimmune products. These are over an FC region, which is shown in the green, but again producing bispecificity with the red and the blue Solamer domains, targeting different disease molecules. And here again, another level of flexibility is that we're incorporating small molecules to make drug conjugates. But really where these domains stand aside from others is that you can link them together to form bispecific or biparatopic domains and keep the molecule really small. So the final product is a fraction the size of a whole antibody. But as a result of that, it's cleared very rapidly from the body. And so EO6 was designed to bind albumin to be fused to other products, such as our own Solamer domains, to extend the half-life, to enable that therapeutic index, that Cmax, that important efficacy to last longer in the body. And we've used that particularly in our oncology products, as you see here with the radiopharmaceuticals. Endure itself was a success story. We had already isolated it by the time this funding started, but we couldn't show whether or not it, as a shark-derived molecule, would elicit any immune responses, but more importantly, could it extend the half-life of other fused products? And here I'm just exemplifying a few options when it comes to those types of new drug modalities. So we can use it to extend the half-life of our own Solamers, as we're doing with our radiopharmaceutical products, but also you can fuse it to single-chain FVs, which are the smallest binding part of antibodies. Down there in the bottom you can see a growth factor fused space Solamer product, again showing that EO6 can extend the half-life of it. And up there in the top right you can see drug conjugates, so again bringing together that small molecule and biologics together, really to create dual functionality but also extension of half-life in the serum as well, so multiple different added on advantages. And EO6, as you see there from the top, we have anointed it as Endure, as it endures in the body. So the project deliverables from the Follow-on Fund were multiple-fold. One, again, these are shark-derived, and if you have any biologic which is not from a human source, you really have to assess it for potential antigenicity. What I mean by that is when it's injected in the body, will it elicit an immune response, which would result in neutralization and potential clearance of that drug. The other big question was, can we fuse EO6 to other molecules, and will EO6 still work? And will those other molecules still work? So that was a critical part of that as well, was looking at the design, the molecular design of these final products. Manufacturability. If you can't make a biologic, you will never get it into the clinic. So looking at that scale up during this project as well. And of course, ultimately, does it do what we want it to do? Can it extend the half-life of other products in the body? So ultimately, this incredible funding enabled us not just to show that EO6 had no immunogenicity, but it enabled us to really bake that into our technology platform, which resulted in learnings for other Solamers which are isolated against disease targets. So it expanded our ability to fast-track humanization throughout our platform and product pipeline. It enabled us to do a licensing deal, which is really important for a small company. They licensed Endure to extend the half-life of a particular molecule that they were interested in. And ultimately, while we were under the umbrella of the University of Aberdeen, it enabled us to build an investable, compelling data package, which helped ultimately securing funding for the company to be spun out from the university in 2016. That was when Elasmogen was born. So Endure is now an integral part of our pipeline. As I said before, it's actively being used in our therapeutic products, particularly in our oncology pipeline around the radiopharmaceutical targeting molecules. And the company itself has gone from strength to strength, which really just exemplifies the importance of this type of funding. When you have something which is ready to be translated, it's really important to focus on that and to try and build a company value around it. Lessons learned? Well, as I said, this type of funding is critical when we get to this stage of commercialisation, that translatable piece to enable us to do the kind of work that you need to secure investment. But beyond that, I have personally learnt an awful lot. I've learnt an awful lot about the commercialisation of science, what matters, the focus on the end point, so the unmet need. Never forget the patient, because this is a long and winding path, and it takes a long time. And you need the right people around you at those different stages as well. So I cannot do this by myself, and so I would like to finish by thanking the team around me, past and present Elasmogen team, but very, very much UKRI and the Follow-on Fund team, because they played a critical role in the creation and the success of Elasmogen. Thank you.
The examples that we have presented so far, including Elasmogen, are highlights from a recent independent evaluation of BBSRC's translational funding. BBSRC has recently published a series of studies that explore our translational and collaborative research investments over the last decade. These studies complement the economic impact assessment of BBSRC attributable spinouts, which was published in 2024. And together they build towards an understanding of how our support is working. The Follow-on Fund is a critical component within BBSRC's translational ecosystem, and the investment often enables research teams to connect with other public and private funding. We usually support an active portfolio of about 50 projects from universities and institutes across the UK. The projects reflect the diversity of the nation's strengths in the biosciences. While health has traditionally been a significant focus, there's been a notable shift towards agriculture, livestock, and food systems in response to evolving sector demands. In the most recent rounds, we've observed a strong increase in projects related to industrial biotechnology and engineering biology. The evaluation of the translational portfolio found that the projects supported by BBSRC have added an estimated £192 million in GVA to the UK, with a future projected overall impact of £652 million. The analysis also indicated a net return of £9 for every pound of public funding invested over the most recent 10-year period considered by the evaluation, demonstrating a clear and positive contribution from the biosciences to the nation. The spin-out evaluation identified at least 450 new spin-outs that can be attributed to BBSRC funding, and estimated that the net GVA contribution could grow to £7 billion over a 20-year period. The spin-outs were connected with a broad range of industrial sectors, reflecting the pervasiveness of bioscience and how research is able to contribute to challenges facing businesses across the entire UK economy. We will now continue with another case study that explores how the Follow-on Fund has enabled progress from discovery to impact.
Hello, I'm Anastasia Callaghan from the University of Portsmouth, and I'm going to be presenting my BBSRC Follow-on Fund work, which is from RNA research to RevoNA Bio Ltd. It's our spin-out story, which was enabled by BBSRC Pathfinder and Follow-on funding. I was the academic PI on both the Pathfinder and Follow-on Fund grants, and I'm co-founder and CSO for RevoNA Bio Ltd. Dr Charlotte Henderson was the postdoc on these awards and is also co-founder of RevoNA and its CEO. Our Pathfinder and follow-on funded work focused on translating RNA research into a commercial platform for high throughput analysis of RNA interactions. So this case study showcases how BBSRC funding was central to every step of that translation process. So our project started with a problem that we faced in our research. That problem was how to study RNA interactions at scale. And my first independent academic grant, which was a BBSRC New Investigator Award, enabled those foundational discoveries to be made. But translating that research into something usable more broadly required targeted support. BBSRC's Pathfinder Award gave us the first opportunity to explore commercial potential and develop a business case. This was followed by 2 follow-on fund grants that enabled us to build, refine, validate a novel platform capable of addressing a real-world unmet need. So thanks to this phased funding, we secured a granted patent in Europe and the US, and we attracted industry partnerships and ultimately secured private investment and launched RevoNA Bio Ltd, which is a spin-out company working in therapeutic discovery through exploring small molecule drug interactions with RNA targets relevant to disease. In recent years, RNA has become a major focus in biotechnology, but methods to study its interactions have lagged behind, and this was our scientific problem. So our early research addressed this unmet need. So the market opportunity therefore became clear. Biotech and pharma companies urgently needed these better tools for RNA analysis. But the route to impact was unclear, particularly for us at a university with limited recent commercialisation experience. So the Pathfinder and Follow-on Fund Awards were not just about advancing the technology. They gave us structured space and support to build our IP, test our model with industry, and prepare for commercialisation. So the Pathfinder Award in 2010 enabled us to test the commercial waters. With positive market validation, we moved into our first Follow-on Fund Award in 2011. And that's where we proved the technology's robustness, and we filed a patent on the technology, and that was in 2012. So our second Follow-on Fund Award was in 2018, and at that point we focused on a high-value application in RNA drug discovery. And that allowed us to partner with a therapeutic biotech company as well, so we could validate our system in a real-world setting. So these Pathfinder and Follow-on Fund schemes aligned perfectly with some complementary support that we got from Innovate UK's iCure programme and BBSRC's Enterprise Fellowship, which enabled us to take the translation of this work actually into a commercial venture. But ultimately it was this follow-on funding that actually made the translational journey possible. So on to the outcomes. Well, the Follow-on Fund Awards helped us to patent the technology, and these patents have now been granted. The awards also enabled us to move from academic prototypes to validated industry-ready technology and to test the system with commercial partners. So this created a platform with broad utility across biotech and pharma, and ultimately led us to spin out the company RevoNA. So RevoNA is the University of Portsmouth's first spin-out in over 20 years. And importantly, this demonstrated that with this critical BBSRC follow-on funding support, a university like ours with limited commercialisation infrastructure could successfully translate its research. RevoNA has now been fully operational for the last couple of years, having raised approximately 1.8 million in grant and equity investment. It currently employs 7 staff and has a strong scientific and commercial advisory board. Currently, RevoNA is a data-generating platform biotech company focused on enabling faster, affordable, RNA-targeted drug discovery. We're working with partners so they can access our technology to conduct RNA-targeted drug discovery at scale. So RevoNA is amassing a rich dataset of RNA small molecule interactions, and these underpin our next steps to build AI-powered tools. Ultimately, our journey shows what's possible, as our success is all built on the foundations laid by our BBSRC projects. So in terms of lessons learned and my advice, Well, if you're serious about translating your BBSRC research for impact, then the Follow-on Fund scheme is ideal. In my case, it gave us the opportunities to test not only the science, but the commercial case as well. I'd really recommend working closely and early on with tech transfer and external advisors, as well as industry contacts. And if, like me, you're at an institution that lacks a spin-out legacy, then I feel the Follow-on Fund model can be the launch pad for spin-outs. It certainly enabled us to create an investable platform, a strong team, and ultimately spin out a company to secure impact from our BBSRC funded discoveries. So for us, BBSRC's Pathfinder and Follow-on Fund schemes were the backbone of our journey. They enabled us to go from pipette in the lab to patent, prototype to partnership, and ultimately from our academic papers to a spin-out company. RevoNA stands as proof of what long-term research translation can look like, especially when funders back you at every step. BBSRC's sustained support gave us not just the foundations to create something, but real ability to make a difference for society. And we're hugely grateful to BBSRC for their support. Thank you so much for listening, and I hope you've been inspired to explore the Follow-on Fund opportunity further. Good luck!
Each year, the Follow-on Fund opens for new applications in January and August. We attract proposals from leading researchers who are exploring some of the most exciting innovation opportunities. The Follow-on Fund progresses our vision to advance the frontiers of biology for a healthy, sustainable and prosperous future. We invest in bioscience researchers as they progress their work towards impact. This year, 52 proposals were received and assessed by our independent panel. The first set of applications to be assessed this year were from the second round of 2024. We received 24 applications, and following the assessment process, 6 of those projects were awarded funding. Altogether, this represents a £3 million investment in bioinnovation, with projects starting in the summer of 2025. Each application was evaluated not only for its scientific and technical merit, but also for its potential economic and societal benefits. Our aim is to ensure that the projects we fund deliver significant added value, driving impactful innovation across the bioscience sector. The successful projects represent diverse areas of bioscience, reflecting the potential for impact across multiple sectors. We were delighted to support the projects in this round, as summarised in this slide. At UCL, researchers are developing a new clinical platform for currently undruggable targets. Meanwhile, at Manchester, researchers are advancing diagnostics with isotropic sugars. In Nottingham, leading researchers are exploring the potential for new compounds for veterinary coronaviruses. In the University of East Anglia, varieties of potatoes are being developed, utilising precision breeding with novel traits. A team at the University of Liverpool is innovating to improve pest control. And finally, researchers at Imperial are seeking to commercialise their research, which we will hear more about in our next presentation.
Hello, we would like to present our BBSRC Follow-on Fund project entitled Commercialisation of a Therapeutic Bacterial Vesicle Engineering and Manufacturing Platform. My name is Dr Richard Kelwick and I'm a Senior Research Associate in Professor Paul Freemont's group.
Hello, I'm Dr Alexander Webb, a Senior Research Associate in Professor Paul Freemont's lab. Bacterial outer membrane vesicles, or OMVs for short, can be used as vaccines to protect humans and animals against a range of diseases.
Thank you, Alex. That is why we are developing our Omega platform to make it easier to engineer and manufacture OMV-based vaccines, including those to help protect poultry against avian pathogenic Escherichia coli, or APEC for short.
Every year, APEC infections devastate poultry farms, causing stunted bird growth, fewer eggs, and can even wipe out entire flocks. Avian pathogenic E. coli infections in chickens, turkeys and ducks impacts over 30% of US commercial flocks and globally causes hundreds of millions of dollars in industry losses every year. APEC-infected birds suffer from severe diseases that veterinarians call colibacillosis, leading to systemic health issues and mortality rates of up to 60%.
Because of this, that's why we are developing our Omega platform to generate these bacterial outer membrane vesicles. A part of this as well is, despite the huge impacts that APEC has on the poultry industry and human health more widely, the current solutions to protect poultry against APEC are ineffective. For example, antibiotics are decreasing in use due to, of course, antibiotic resistance. There's also a general trend in terms of regulations and other policies minimizing their usage. But also farmers themselves know that poultry treated with antibiotics can have side effects which impacts their health, their meat production and also their egg production. Unfortunately, the currently widely used live attenuated vaccines, such as those developed by Zoetis and Chemin, whilst fairly effective, are not always fully effective, especially against changing strains and for cross-strain protection. Additionally, farmers also want rotation products. This is because farmers are rightly concerned about resistance emerging against either vaccines or antibiotics, and so they want different solutions to protect their flocks moving forwards. Therefore, our proposed approach is to make it easier to develop ONV-based vaccines, and so we have developed our outer membrane vesicle engineering and manufacturing, or Omega platform, to make it easier to create better and more scalable methods for manufacturing new kinds of therapeutic or vaccine ONVs. In our project, we are using Omega to design, engineer and manufacture across different scales a panel of candidate ONV-based APEC vaccines. We will also characterize our vaccine candidates and use in vitro screening to determine their effectiveness. And of course, we want to expedite a responsible translational pathway for our Omega platform. And we are pleased that we have collaborators and supporters in our project, including but not limited to Symbicity, the London Biofoundry in Imperial College, and the Future Biomanufacturing Research Hub at the University of Manchester. So, our Omega platform. Our Omega platform allows us to create bacterial outer membrane vesicles that are engineered from standardized bacterial strains. They allow us to display multiple vaccine antigens, and we can also have designer protein cargoes. Our platform is modular and standardized, meaning we can create many vaccines from the same platform and also many kinds of therapeutics. We believe our approach is novel in that we can enrich for our engineered ONVs, which distinguishes from other technologies which often isolate a mixture of ONVs or EVs produced by different cell types. And our approach is also scalable. And as part of our project, we want to go from our current small scale in the lab to a higher scale using a bioreactor system. And in discussion with Imperial Enterprise, we believe our approach is patentable. There are core innovations as part of our platform. In terms of our future vision and expected outcomes, as part of our project, of course, we want to demonstrate our Omega platform and its ability to create many different therapeutics and vaccines. Of course, our focus is on the development of poultry vaccines, and so we want to demonstrate that engineering and manufacturing at different manufacturing scales and to test our candidate poultry APEC vaccines for their efficacy in vitro. In terms of long-term impact, we want to continue our engagement with industry, and this builds upon our participation in the BBSRC iCure program. And we believe that we can file a patent for our Omega technology, and we have support from Imperial Enterprise to do this. And in the long term, we believe there's scope for licensing of our technology and platform and/or potential spin-out.
The first round of 2025 opened in January. We received 28 applications and 6 projects were awarded funding, with a total of £3 million invested in bioinnovation. Most projects started in the autumn of this year, and we're pleased to continue supporting cutting-edge research with significant potential to create real-world impact. We received a strong response to this round, with engineering biology and the circular economy featuring particularly strongly. New projects will explore innovations in textiles, feedstocks and intermediaries. Researchers will also focus upon improving human health by addressing biting insects and improving resilience to climate change in the context of food security. We now move on to a series of case studies from this round.
Hi, my name is Emily Flashman and I'm a PI at the University of Oxford in the Department of Biology. I'm really excited to tell you about a project I'll be launching soon along with my co-workers Mikael and Rebecca called FloodAlert. This project builds on some of our previous research looking into the mechanisms by which plants sense when they get flooded and how they adapt to that flooding stress. And we want to use that knowledge to develop biostimulant treatments that can help make plants more resilient to flooding. Flooding is, of course, really damaging to plants and is a major cause of yield loss in crops. And this is increasing globally with climate change. This is expensive for farmers, both in terms of the yield loss, but also in terms of the loss of the land that they can reliably use for farming. It also costs a lot for governments in terms of compensation strategies, and that's also not considering the cost to consumers for the increase of prices in staple foods. There's currently no treatment targeted to protect crops against flooding, and so we now want to use a research-backed approach to develop an effective treatment that can help protect crops against the damage caused by waterlogging and submergence. And we're going to develop these treatments so they can be applied as a biostimulant at the seedling stage of winter crops, so offering them protection against flooding at a time when they're particularly susceptible to that sort of stress. Plants are able to sense when they're flooded due to a reduction in oxygen availability. This is also called hypoxia. Under hypoxic conditions, such as when their plants are flooded, transcription factors called ethylene response factors, or ERFs, are stabilized and are able to upregulate genes which allow the plant to adjust to the flooded environment through a metabolic reconfiguration. In other words, converting from aerobic to anaerobic metabolism. We have found that the enzymes called plant cysteine oxidases, or PCOs, are conserved enzymes which use oxygen to trigger the degradation of these ERF transcription factors under normal oxygen environments. It's the reduced activity of these PCO enzymes when plants don't have enough oxygen available that results in the stabilization of the ERFs and temporary plant survival of submergence. It's known that stabilization of ERFs can improve their survival of a future low oxygen event. And this can be artificially induced even when plants are not flooded in what is known as a priming event. We have shown that reducing PCO activity via competitive inhibition is an effective way of achieving such a priming event. So we're going to build on this knowledge to develop potent and biosafe activators of plant submergence tolerance, focusing, as we say, on winter wheat. Scientifically, we will develop a toolkit of biostimulants using a pipeline of screening platforms comprising established and rapid in silico, cellular, seedling and protein assays, which will identify a suite of potent PCO-specific biostimulants. We will then test these in an in-house winter wheat submergence assay for their ability to promote survival. So far, so academic, but of course this is an opportunity to translate our findings into something that's practically and commercially valuable. We have already conducted market research which suggests a strong need for products that can secure winter crop sowing against flooding risk. So, alongside the technology development, we will therefore conduct activities that will ensure that we're ready to exploit our top hits in the field as soon as possible. This includes securing intellectual property rights, making sure our hits are aligned with agrochemical regulations in the UK and worldwide, worldwide, and ensuring we're able to produce our HIT compounds at scale and in a formulation that can be applied to farmers' fields, preferably in spray form in combination with other treatments using existing machinery. This will help keep costs low for farmers. These activities will require us to secure project partners. So in the early stages of the project, alongside our initial HIT refinements, we will be seeking productive partnership opportunities. By the end of our follow-on fund grant, we will have a suite of products that are produced at scale and ready to apply to field trials. Assuming we see a successful outcome, we will then select the most appropriate route for market delivery and will establish a spin-out company with which to deliver this. We'll also aim to expand our product range, including protection for different crops. Overall, the outcome from this grant will be the delivery of a crop protection product that can give farmers confidence that winter-sown crops can have genuine protection against flood damage and therefore increase yields in the UK and beyond.
Hello, my name is Jinsong Shen, Professor of Textile Chemistry and Biotechnology at the Montfort University in Leicester. Here I'm to present our recent funded BBSRC Follow-on project called Scaling Up Bioprocesses for Fibre Separation of Mixed Textile Waste for Circularity. This project will be led by myself and project co-leaders Dr. Chandra Prajapati from Loughborough University and Dr. Edward Smith from De Montfort University. This project is supported by 7 industry partners: Roberts Recycling, Camilla Fabrics, Woolmark, Fox Brothers, Meta, Wilson North, Dye Recycle. The project is expected to start on 1st of February 2026 for the duration of 2 years. The project aims to tackle key challenges in the textile sector by focusing on the recovery, recycling, and the reuse of mixed textile waste to promote circularity. We know the rapid growth of textile consumption demands greater use of resources and enormous amounts of energy and water for producing virgin materials and processing into textiles. This results in the depletion of natural non-renewable resources, contributes significantly to the carbon emissions, which is unsustainable. From a European Parliament report recently published, the EU generates 12.6 million tons of textile waste per year. Less than half of user clothes are collected for reuse or recycling, and only 1% of user clothes are recycled into new clothes. Blended fabric made from different fibers or mixed textile materials are currently difficult to recycle for producing high-value fibers for remanufacture. Blended fiber must be separated before the individual components of the blend can be recycled. This cannot be done simply by mechanical separation, due to a lack of innovation to enable effective separation of different fiber components. Blended textile waste often ends up in either landfill or incineration. Our initial BBSRC funded project, which finished in January 2025, responded to the challenge of current problems related to the recovery, recycling, and reuse of blended fabrics or mixed textile materials. The project successfully developed the enzyme-based biotechnology for separation of fiber components from wool blended fabrics, recovered dyes from waste wool materials, and recovered synthetic fibers, flax, hemp fibers from wool-blended textile materials without damage, offering opportunity for the components to be processed for reuse. This follow-on project builds on enzyme biotechnology we have developed from our previous project. It will explore the potential for scaling up the biotechnology for the separation of different fiber components from wool-blended fabrics and the recovery of dye from waste wool component fibers. The project will involve the design of prototype bioreactor, specially used for enzymatic fiber separation and dye recovery, and the optimization of enzymatic processes for scaling up. Our follow-on project is highly collaborative, involving 7 industry partners. This diagram shows the project approach. The project will focus on wool-blended waste textile materials. Project partners Roberts Recycling, Camera, Fox Brothers, and Woolmark will provide post-consumer wool-blended garments and manufacturing waste. Waste wool blended fabric will be rag opened and or shredded into mixed fiber form or small pieces of fabrics. Camera has a rag opening system built by Wilson Knowles for mechanical recycling of wool fabric into wool fibers, suitable for respinning into yarn for production. The waste fabrics will be tried for opening at Camera. The rag opening system could be modified to be more suitable for recycling of specific wool blended fabric if required. In the project, prototype of bioreactor will be designed for enzymatic processes for separating mixed fibers from waste materials, whereas the bioprocesses are optimized for the potential scaling up. The project partner Dye Recycle will provide knowledge and expertise for designing the reactors and for recovering dye from waste textile fibers to produce recovered dye powders. In addition to centrifugation, the industry partner Meta will provide different filtration systems for separation of wool fragments or protein from the bioprocess red liquor and collection of recovered dye. Industry partners will explore the remanufacturing of recovered fibers and the reuse of recovered dyes for fabric dyeing and printing. The resulting textiles will be tested and assessed for the potential of commercial applications. We are aiming to achieve a fully operational system for textile recycling and reuse of wool-blended or mixed textile waste materials, including collection of waste textile materials, sorting, rag opening, and shredding, bioreactor system for enzymatic fiber separation, remanufacture of recovered fiber and dye for commercial use. The project will provide critical data on environmental impact and economic benefit of the outcomes. The project will develop and deliver strategies for commercial implementation and the marketing of recovered fiber and dyes and potential applications.
I'm Dr. Benjamin Lichman from the Centre for Novel Agricultural Products at the University of York, and it's my pleasure to have received follow-on funding from the BBSRC entitled Enzymatic Route to Insect Repellents from Bio-based Feedstocks. Stinging and biting insects are problematic for human health. Mosquitoes are well known as vectors of serious diseases such as malaria, dengue, and Zika. They are responsible for over 700,000 deaths a year. Other stinging and biting insects are also problematic. Ticks, for example, transmit Lyme disease, and black flies transmit leishmania. Furthermore, our heating climate is causing an influx of invasive species, moving away from the equator and towards the poles. The nuisance of biting or stinging insects is likely to get more acute in temperate climates like the UK over the next few decades. One major way we protect ourselves against these threats is through insect repellents. The most widely used repellent is DEET, which, although effective, has an unpleasant odor, dissolves plastic, and is perceived of poorly by some parts of society. DEET is derived from fossil fuels and synthetic chemistry, so it is not part of our vision for a circular bio-based economy. We think there is a key gap in the insect repellent market for a different type of product. Winning just a portion of the global market would have a massive impact. The total market size was estimated to be nearly $4 billion in 2018 and expected to expand, especially as global warming increases the range of tropical diseases and resistance to current repellents develop. Plants from the genus Nepeta, such as Nepeta cateria, catmint, have natural insect repelling properties. In fact, cats have evolved to be attracted to catmint, so when they rub the plant, some of the catmint oil is transferred to the cat's fur and protects them from biting insects. The active insect repelling chemical from catnip or catmint is nepetalactone, which, whilst effective, is unstable, has an unpleasant odor, and can sensitize skin. The similar chemical, dihydronepetalactone, or DHN, is a minor natural product of catmint and has similar repelling activity but is more stable, has a pleasant odor, and no evidence of sensitization. We think this molecule has great potential as an insect repellent. Our project partners, Entomol Products LLC, a US-based company, have developed the product hydrogenated catmint oil, HCO, which has DHN as an active ingredient and is registered and marketed for use as an insect repellent in the US. They have developed a farm-to-formulation pipeline wherein they obtain steam distilled catmint oil from farmers, hydrogenate it using proprietary conditions, and then formulate it into products. Their consumer products are marketed under the brand NIPT, currently for sale in the US. Although this is a great method, catalytic hydrogenation is costly, uses excess solvent and precious metals, and is ultimately not part of our vision for a circular bio-based economy. Therefore, we are looking for an alternative, biological-based approach to make hydrogenated catmint oil. Indeed, we have successfully found enzymes that can catalyze this reaction, taking catmint oil and converting the nepetalactone into DHN. This is a great proof of principle, a discovery originally funded by the High Value Biorenewables Network. But now, in this follow-on fund, we need to optimize the process to compete with the catalytic hydrogenation. We will be using enzyme and chemical engineering to boost conversions and rate and capacity of the enzymatic process. Our vision is a fermentative or bioreactor-based process that takes crude catmint oil and converts it to enzymatically hydrogenated catmint oil to be formulated into repellent products. But we are also working towards plant-based processes to produce DHN from sunlight. Eventually, we aim for our technology to be a reliable way to produce insect repellents, protecting us from mosquitoes, flies, and ticks. And furthermore, we and our environment will be protected from the negative aspects of DEET production. As the threats from biting and stinging insects increase, our technology and its products will fill a gap in the market for a plant-derived, pleasant, and effective insect repellent.
We would like to thank all of our speakers who have so generously recorded presentations for us. The fund is designed to offer flexible support for researchers to partner with businesses of all different scales and sectors. Previous and current award holders are engaging with biopharmaceutical developers and manufacturers, agri-food businesses from field to fork, technology developers, including research equipment manufacturers and providers, producers of bulk and specialty chemicals, as well as consultancies, utilities and third sector associations. Businesses that participate as project partners on BBSRC Follow-on Fund awards co-invest alongside BBSRC to commercialise research into new products, processes and services. The fund uses a framework that enables businesses to flexibly access valuable intellectual property based on their level of contributions, whether those be in cash, in kind, or a combination of both. Project partners who contribute 10% of the total project costs can gain a first right of refusal to negotiating access to IP, helping to de-risk early-stage business or investor engagement. Partners who contribute 50% or more can secure pre-negotiated access to IP, ensuring a strong stake in the project's success and future commercialization potential. For academics aiming to spin out a new business, they are given the flexibility by exempting empty shell companies created solely to commercialize the project's IP from contributing to project costs. By offering flexible options for both academia and industry, we aim to ensure that every team can choose the commercialization approach that offers the greatest potential for success. We hope you've enjoyed the presentation so far. We've really enjoyed being able to showcase some of the exciting new projects that we've been delighted to support this year. The fund has built a community of leading researchers and innovators who have collaborated together to unlock the potential of bioscience. The projects create partnerships that not only generate new intellectual property, but which also create enduring relationships with strategic value across businesses to open new commercial opportunities and remove internal constraints. In doing so, the Follow-on Fund is able to de-risk innovation across the biosciences in a variety of ways, including by decreasing the time and cost of product development and manufacturing, and addressing risks associated with the needs of both customers and regulators. Participating in Follow-on Fund projects has created direct commercial and competitive value for businesses and has enabled leading-edge new products, processes and services to be brought to market in the UK. As we draw this part of the webinar to a close, we would like to direct your attention to our next launch event. We would love for you to join us as we explore the first round of 2026 in February. The launch webinar is primarily for potential applicants and will describe the process of making an application. However, anyone who is interested is always welcome. The launch of the next round of FOF will be advertised first on the UKRI Funding Finder, where you can find information about all of BBSRC's funding, including support for knowledge exchange, translation and commercialization. I'd now like to take the opportunity to highlight some other funding and resources available from BBSRC. If you would like to know more about the awards made by UKRI, then summaries of all existing projects are available online through the Gateway to Research website. You can search directly for projects that relate to your discipline, technical area, or business interest. There are a variety of other funding routes for partnering, including our IPA and LINK programs. These are part of responsive mode and are always open for applications. These programs enable collaborative R&D projects with a value of up to £2 million and are a fantastic option to advance partnerships. The Follow-on Fund is designed to interface with responsive mode, and so these awards may be a more suitable option for ideas that are not yet mature enough for a FOF award. We are delighted to be partners in the iCure programme, which is another option to develop your idea. iCure is a UK-wide bioscience pre-accelerator that trains, funds and supports teams led by university researchers to progress from the lab by validating their business ideas in the marketplace. iCure is open to all researchers and technical research staff at any career stage and at any UKRI eligible research organisation. We increasingly see teams progress through iCure towards FOF, either within their project or as part of developing their commercialization strategy. Strategic support is also available for venture. And as an example, the BioBic program, which is a partnership with STFC, supports early-stage biotech startups that are built on leading bioscience research to bridge the gap between science and market to achieve their full commercial potential. Startups participating in BioBic receive a package of support, including funding, access to incubator space, and coaching, all to accelerate technology development and attract private investment. The program is just one example of how we're working to develop the bioinnovation ecosystem. We connect the bioscience community by funding networks and hubs that bring together researchers and innovators to stimulate collaboration and build capacity in areas of strategic importance. These forums explore a diverse range of subjects, from healthy aging to food and nutrition, biofilms and novel proteins, through to industrial biotechnology, new tools and technologies, and areas where the biosciences interface with digital and engineering disciplines. These communities are fantastic ways to connect and to explore novel opportunities and ideas. Another way to find partners is to use the Confer tool. Confer is the UK's smart matching innovation tool and is supported by UKRI alongside the National Centre for Universities and Businesses. It's a fantastic way to find new resources and new opportunities. We would also invite you to connect at our annual conference. Sparking Innovation is centred on bringing together our community of bioscience researchers and innovators to explore knowledge exchange, translation and commercialisation. The event is a highly interactive format featuring networking, pitching and panel discussions over 2 days as we aim to spark innovation. We have had very positive feedback from delegates who return year on year to learn more about the opportunities to take their research further. Attendance is free and registration will open in January 2026. Sparking Innovation will be advertised first in our Innovation Bulletin. The Bulletin is a dedicated newsletter that focuses on opportunities related to knowledge exchange and commercialisation. It's where we advertise events like Sparking Innovation and also all of our translational funding opportunities in one place. I'd like to finish by thanking all of our presenters for taking the time to record such fantastic impact case studies and forward-look presentations. The Follow-on Fund has an entire community around it that comes together to create opportunities for investment. We rely hugely on our grant holders, our applicants and our assessment panel members to continue investing in the most exciting new bioscience research and innovation. We thank you for being part of our community today and we hope to see you at future events.